Good evening, my dear students. You are all warm. Welcome to the wonderful session of the biological classes. Today, what we are going to discuss about the remaining part of the biotechnology. Particularly, last class, what we have discussed. Okay, so already you know very about DNA cloning, how the DNA molecule clone into the other organisms. Before that, what are the experiments involved in it? That things we will discuss. First of all, cloning experiments involves isolation of the gene of the interest. That is very, very important. Which gene we are going to amplify by the process gene cloning method, which gene is very, very essential. That things you must isolate from the different sources. Already you know a little about maximum living organism only, the plant as well as the animal sources is there, particularly the extra chromosomal body is there. Okay. So good evening, my dear students. Today we are going to discuss about the
Good evening, sir.
Good evening, Kanna. Good evening, Ba. So today we are going to discuss about the gene cloning and their steps. What are the steps involved in the gene cloning process and how it will be? DNA molecule is also transferred from the one generation to another generation. Okay. These things today we are going to discuss. First of all, you should know. So what is by gene cloning? Already we discussed this part. The term gene cloning can be defined as the isolation and amplification. Isolation means it may be isolated from the different species, whether it may be animals or plant species. Okay, sometimes it may be same family, sometimes it may be the same genus and species also. Okay, that after that isolation of the selective DNA molecule, and also undergo the amplification process. What is the meaning of amplification? Amplification only done with the help of the PCR. Already we discussed this, indeed, polymerase chain reaction. But in this category, gene cloning methodology is also one of the amplification technique without any laboratory work. It is also laboratory, but it's not used as a, which one is that there is no any prime, that things no need for the amplification process, because it is done by where, by the microorganisms such as E. coli and phage, or some of them are having this property. See here, and after that amplification of an individual gene sequences by inserting, that is very, very important. Inserting means the course is essential one. Of that sequences into a bacterium or other cells maximum. What is that? Bacteriophage. Okay. Other living organisms. Take an example of iron element did, the Dali, 1996 is one of the cloning methodology. Okay. And successfully he did. Finally, after 6 months, what happened? The goat was died. What is the reason behind it? Because due to the lack of immunity. So that gene is not responsible for the disease within the cloning byproduct. Cells where it can be replicated, that is very, very important. First of all, cloning experiments involves the isolation of the gene of interest, which genes responsible, whether it may be color or whether the disease resistant, whether they are antibiotic resistance, any other things is also, whatever it may be, sexually transmitted characters. Okay, suppose if it is related to our what is that? Color blindness. Okay, what are the sex-linked inheritance, the gene-related ones? That things is also comes under this heading. Okay, so go to the next one is how it will be isolated. Already we discussed that in the last class. Once again, I remind you. The isolation of the gene of the interest gene, we are going to isolate. How a fragment of DNA, so gene is insert. Okay, that gene is to be clone is incorporated into a small replicating DNA molecules called a vector. Vector means you know about that. Which one is isolated? It is nothing but the sample 1. Which one we are going to introduce? That is sample 2. Okay. The vector with the incorporate gene is called as recombinant DNA molecules. It is simply called as, what is that, R, so DNA. Okay. The rDNA is nothing but the recombinant DNA molecule. Sometimes they may ask the question in expansion aspect. Okay. So next, after that, what happened? After the getting the recombinant DNA molecule isolated, what will have to do? The recombinant vector is introduced into the host cell by transformation. Transformation is nothing but one gene transferred to the one organism into another one. Okay. But here the vector is introduced into the host cells. That means after that, two DNA molecule united together form as a vector. That vector is introduced into the host cells. How it will be enter inside the cell, there are some technique is also there. Gene gun, okay, electroporation, and so many things is there. Okay, they are all are very minute quantity. Already we talk about that things. Already we talked, isn't it? So very minute quantity, that is 20 microliter, other less than 20 microliter, so 2 microliter, like that. That component will be easily inserted. That is also transformation takes place from the outside to inside the cell. Okay, so the cell that have the acute recombinant DNA molecules are selected. The recombinant DNA molecule is multiplied within the host cell to produce a number of identical copies of the cloned genes. Last class we saw it, that one. Okay, see then already I saw it. Again I will repeat for you. Okay, so this is a consider a gene again and again, a sample, otherwise called as a, what is that, strain. Okay, a sample. So another one is considered as a B1. Okay, another one is considered as a, this is B sample. Okay, so B sample is there. So inside already you know, but there is a nucleic acid, otherwise called as extra chromosomal body present in it. That thing's also already in our report. Another one is also very, very important one. So in this category, you have to select another one, then a bit better for you. Both are same. Okay. So this is the another one. So both are what happened, you know, united. Okay. Both are united together. How it will be already, you know, report. The gene will be selected gene. This is considered a selected gene of the this part. Okay. So it is also incorporated with the another gene sample, another gene. Okay, so it's also incorporated with the another one. So already you know this is the A gene. So this one is called as B gene. Okay, these two are combined together. We will get the vector. This part is called as, what is that? Vector. Okay, vector. Okay. So after that, that vector, after the recombination of the two GNA molecules, this one, which is, this part is a selectable or interested one. Interested. Okay, this gene is united with the help of the molecular system already in our restriction enzyme, also used for the cutting process and also linking process. Which one you know? Ligases, DNA ligases also used. After that, what you have to do? You are going to introduce the host. Very, very important. Very, very important is host is that. Okay. This after this setup, okay, they are going to introduce the host. This is the host. Consider as a host. Okay. Post maximum E. coli, because E. coli is very easy to handle. Genetic engineering they were used. Suppose take an examples in prokaryote. Okay, in prokaryote concept, E. coli is better. Okay, so this setup is completely set inside. It's inserted with the help of microporation. How it will be entered? It's also loaded with the help of the electric gun. Okay, so after that, what happened? No, automatically it will be enter inside. How it will be? No, this is like this. So it will be enter like this. So pour it. It's also inserted with the help of the gun, electric gun, or whatever it may be. What are the equipments that were used for the this thing? This things is also introduced inside the host. This is the host. So there is a extra chromosomal body of the both the genes. These genes. So this part is very, very important. This part is or this part. This part is very, very important for the replication process. This is the selective one. Selective. Okay. Selective. So what is the meaning of selective one? You know, what is the reason behind it? Selective. Because it is essential character. Essential character is the, so it replicates itself inside. What happened? No, so many replication takes place. So many things. So many. That is called as amplification without any, what is that? Any techniques. Okay. So how many is there? Inside there is so many molecules. So after that, it may introduce into the where? The plant or animals. Plant or animals. Okay. So the plant or animals, they have the ability, whether it may be resistant characteristics or some of the special character, which characters we are selected. Okay. So interesting character, you know, that character, it may be resistant power or cold or tough or whatever it may be. It's also another thing is antibiotic resistance is the disease resistant, pest resistance. So many things is there. They are all introduced into the caused to the plant or animal, what happened to the plant or animal, they are having the special character. Okay, so this is, are you understand, Bob?
Yes, sir.
Okay, dear. Listen here. So this is the recombinant vector introduced the host cell by transformation. Cell and that have acute recombinant DNA molecules are selected. The recombinant DNA molecule is multiplied within the host cell to produce a number of identical copies of the cloned gene. That's why I saw it. Okay. Last slide we saw it, isn't it? So, okay. Next. So what are the steps involved? First one is DNA extraction. The DNA extraction, it may be isolation of genetic material from the cell, whether it may be a bacteria, viral, or animals, whatever it may be. These are the samples. These are the plants or animals or taken. After that, you have to rupture the cell. Then only it will comes out, isn't it? So the center, there is a nucleus. Inside the nucleus, there is a DNA is there. DNA, it has a characteristics. Okay. So that's why. So you have to rupture the cell wall. Then only, not only that cytoplasm bound organelles, cell bound organelles also comes out, isn't it? Endoplasmic reticulum, Golgi apparatus, mitochondria, and dictosomes and ribosomes. Okay, ribosome, rough endoplasmic reticulum, and also smooth endoplasmic reticulums, and also suppose if it is plant cells, taken examples of chlorophyll also there, and also vacuole is also present in it, isn't it? All those things present in it, but we need only the DNA characteristics, DNA only. So we have to rupture the cell wall, then also enter into the nucleolus, and also how it will be, say, rupturing of the cell membrane to release the cellular component and DNA, first point, and separation of the nucleic acid from other cellular component, from the nucleic acid only, because all the component present inside it. We need only nucleic acid only. So you have to separate from the other component, other cellular component. Okay. So we need only the DNA molecules only. After the isolation of the DNA molecule, it undergoes a process called as a purification process. Once again, you have to rupture the cell membrane. That time what happens, other components also comes out. So from that one, we have to isolate the DNA alone. After the DNA separation, what happens, it also undergoes the process called as a purification process. Okay. So that is very, very much. After that, for tissue culture, okay, tissue culture or blood samples incubation, the buffer containing detergent, this is very, very important idea. Very, very important. So sometimes they were used, what are the buffer solution used for the tissue culture method or blood sample, blood culture. Okay. So sodium dodecyl sulfate, this is very, very, SDS is an expansion of sodium dodecyl sulfate, is nothing but the detergent, buffer containing detergent. Don't forget, my dear. Buffer already, when you're adding the acid or base, what happen, the pH, it will not change. Such type of solution is called as what is that? Buffer solutions. Already we know about. Where will you used? You know about that. Electrophoresis already we were used, isn't it? In tissue culture also we were used. And also blood culture. Suppose take an example. Dhanalakshmi. Okay. Hello. Dhanalakshmi, are you in?
Yes, sir.
Okay. Will you please tell me how the COVID virus are cultured? You know about tissue?
Sir.
Yes, tissue culture, you know very well, isn't it?
Yes, sir.
So tissue culture means, the plant or animals, both are tissue, isn't it?
Yes, sir.
How will you grow? With the help of?
Tissue, sir.
Okay, da. What I'm asking, there is a tissue culture means, what is that, they need some medium, isn't it? Culture medium. The culture medium, it contains what is that, there is a nutritive substances present in it, agar as well as other components also. Suppose if it is plant, there is a phytohormones is also very, very important for their culture, isn't it? What I am asking, here, the COVID is also one of the viruses, otherwise called as bacteriophages. How it will be cultured inside the lab only? How they are analyzing the medicines and so on, so the vaccination, that things also, how they are analyzing with the help of the microbes. How it will be analyzed? Any idea?
Don't know, sir.
Okay. Right, anyhow, how it will be, there is a blood culture, very, very essential. Blood means you take a human blood or sheep blood. Okay. After that, it's also sheep blood. It's also containing nutrient substances and also agars also. There, what happened, we are adding the particular microorganism pages, it will easily grow. So the virus or only grow with the blood culture only. That's why they are using in the laboratory for the vaccination process. Okay, anyhow. So here we should know, so what is a buffer system? Sodium toadicil sulfide is usually sufficient to disturb the cell membrane and release high molecular weight. And cell are lysed opened to release the DNA along with the other macromolecules such as RNA, protein, and polysaccharide, and also the lipid molecules. That things already we discussed, isn't it? See that one. Usually, a machine is used to extract the DNA from the cell is called as bead beaters. Don't forget, my dear, how the DNA molecules extracted from the air in the cell. Okay. So with the help of the one of the machine, okay. So that is called as a bead beaters. It breaks the cell and extract the DNA from it. And electric current is used for this purpose. Okay. That is very, very important. How the cells are lysed with the help of the knife? No. Any other instrument, we cannot be able to suck with the help of the syringe. It is not possible. If we have to rupture means we have to use some mechanical support, machines. Okay. So that is called as what is a bead beaters. So as a result, what happened, the DNA molecule isolated or comes out from the cell. So after that, what happened? The bacteria surrounded by a cell wall can be gently broken open by treatment with the combination of lysozyme and ethylene diamine tetraacetic acid. These are very, very important because ethylene diamine tetraacetic acid is one of the calcium chelating agent mostly used for the blood counting, total count, WBC count, whatever it may be in the DMLT lab, they were used for the anticoagulant substances. Here, the EDTA easily bind with the calcium of the bacteria cell wall, so it can be easily ruptured. Lysosome already in our lab, it's one of the organs. It is also called as what is a suicidal bag, isn't it? It has secretes so many hydrolytic enzymes. They act upon the cell wall. As a result, the cell wall is easily broken. Okay, the EDTA bind with the cell wall, so the cell wall loses the ability. The lysosome easily what happened? Rupture the cell wall. So as a result, the cell bound components it will be comes out. It is very, very important. Go to the next one. This can also be achieved by plant or animal tissues with the enzyme cellulase, plant cell, and chitinase is a fungus. Already you know about. Okay. So take an examples in this part, lysosome. See here. Take an examples of any one of the plant. Okay. So any one of the plant, take an examples, any one of the plant. Here also take an example, simple diagram I draw here. Okay. So from this one, what is this? The cell is made up of, okay, this is a cell, single cell I draw here. So inside what happened? There is a nucleus is there, isn't it? The cell wall is made up of what is that? Cell wall. Cell wall is made up of which one, cellulose, isn't it? Cellulose, isn't it? Suppose take an examples of animal cells. Okay, this is the plant. So take an examples of plant cell. Okay, suppose take an examples of animal cell. Animal cell is made up of what is that? Already but the animal cell is also made up of membrane, isn't it? Animal cell is also made up of membrane, but inside what happened, this is phospholipid. Otherwise called as phospho, PHOS, PHO, okay, phospholipids, okay, lipids, because it's animals cell membrane, okay. Inside there is a nucleus, already you know a little bit, there is a nucleus. This is the animal cell. Animals, these are all plant cell, animal cell. Suppose take an examples of fungi, okay. Any one of the fungi, take an examples of fungi. What is that, the fungi, the cell wall is also there. The fungi cell wall is made up of which one, chitinase. Okay, here I will type it, this better for your understanding, okay. See here, fungi means fungi cell wall is made up of chitin, isn't it? Chitin, CHITIN, okay. Chitin, this is very, very important. This is very, very important idea, okay. So here what happened, here the cellulose is there, the enzyme act upon it, what happened, the cell wall is easily ruptured. That cellulose is a carbohydrate, the enzyme is cellulase. Here the animal is there, okay, lipoprotein, lipase. Here chitin, chitinase. So all are enzymes, easily rupture the DNA molecules. What is the nucleus comes out? This is very, very important. Here rupture happened. What is that? Next, what happened, automatically it comes out. This DNA molecules comes out. Here there is a DNA molecule, the otherwise called as nucleus comes out. Along with this, so many microorganisms, so many cell-bound organs also present in. Cell-bound organs already you know, isn't it? See that one? Inside there is so many cell-bound organs also present in it. That is called as, what is that? This is a ribosomes, and another one is called as mitochondria. Okay. Here the Golgi apparatus is there. Okay. And also vacuole is also present in it. Here also same. Here extra, what happened? There is a chlorophyll molecule present in it because it's a plant cell. So the chlorophyll molecule is present in it. Okay. Same as well as this one is mitochondria. Okay. Chlorophyll, ribosomes, endoplasmic reticulum, all those present in it. But what happened, what we are doing, first we have to rupture the cell wall, whether it may be plant, means if the plant cell wall is made up of cellulose, so we have to use the cellulase enzyme, cellulase. Okay, so that is very, very important, my dear. So cellulase enzyme we were used to rupture the cell. So what happened, the DNA molecules comes out, it comes out. That is very, very important. Okay, after that what happened, the DNA molecules comes out. Likewise, the animal cells also, lipase means we have to break this point. So the DNA molecules comes out in this part. In this part, the DNA comes out. Suppose if it is fungi, which samples we are selecting depends upon their sample cell wall, which component is made up of, you have to find out, and then you have to use the particular enzymes. Then only the cell wall is easily broken, otherwise it will not break. Okay, okay, see the fungi. Okay, fungi, what happened, you know very little bit. Chitin is a, very little about that. Chitin is a cell wall component. It is made up of chitin. So the enzyme is called as chitinase. Chitinase. NASE. Okay, chitinase. The chitinase is one of the important enzymes. Act upon the chitin. It is mostly found in the fungi. Okay, these are the very, very important, my dear. Okay, okay. So go to the next one. See that one. Okay, lysosome, ethylene DNA tetra. Already we discussed these things also. See that one. So this is the cellulose is the plant cell, and also the chitinase is the fungi. And ethyl diamine tetraacetic acid, EDTA, it is a chelating agent, calcium agent necessary for the destabilizing the integrity of the cell wall, and it's also inhibit the cellular enzyme that degrade DNA molecules. This is also very, very important, my dear. Okay, so don't forget what are the chemicals they were used for the breaking of cell wall, whether it may plant or animals. Here they were used for the animal cell wall. Okay, don't forget. And second one is called as SD, as already we discussed. Okay, sodium todecyl sulfate. It is also essential for the removal of the lipid molecule and denature of the membrane protein. This is also very, very important. Sodium todecyl sulfate. It is one of the chemical component used for the what purpose? The same process already we discussed in the cell plasma membrane. Plasma membrane is also made up of which one? In lipoprotein. Lipoprotein easily denatured with the help of the which one? Sodium todecyl sulfate. Don't forget. Okay. Next one, in an enzyme protease is used to degrade all kind of protein, whether they are associated with the DNA or present inside the cell. So you all are having the enzyme activity only, enzymology. Okay. Some of the chemical substances used as a, what is a cellular breakage. Okay. Some of the enzyme used for the respective substrate activities. Okay. These are the very, very important. What are the things already here studied, cellulose enzyme and also chitinase enzyme. Ethyl diamine tetraacetic acid is also one of the chemical substances. It is also act as a calcium chelating agent in where, blood clotting mechanism. But here you just note it down. It is also essential for the cellular enzyme that degrade DNA molecule. It inhibits the cellular enzymes. And sodium todecyl sulfide, it is also help to remove of lipid molecules and denature of membrane proteins. And enzyme protease is also very, very important. Okay. It degrade all kind of proteins, whether they are associated with the DNA or present inside the cells. The protein molecule completely what happened degraded. The DNA molecule is also easily isolated from the combined form to free form. Okay. So this is the another point. So next one is the RNA can be removed by treatment with the ribonuclease. Already we discussed these things also. Other molecules can be removed by appropriate treatment. Each one has a specific treatment because they are having own specific molecular weight. Okay, my dear. Okay. So go to the next one is called as after the removal of the protein or DNA molecule will be present at the interphase. Then the precipitation of the DNA is brought about by the cold or chilled ethanol. This is also very, very important. Yesterday we saw it, I think, last class. Chilled ethanol. So then what happened? The DNA molecule undergoes the precipitation process. Okay. So this is very, very important. NEET point of view. It is very, very important. DNA precipitation also done by which chemical component? Okay. The chilled ethanol. That is very, very important. Okay. This can be seen also collection of the fine thread in the suspension. After precipitation, it is centrifuged. DNA is insoluble in alcohol. That's why we are using ethanol. Therefore, it comes out from the solution immediately. Okay. Sometimes they may ask the question true or false. DNA is insoluble in alcohol or soluble in alcohol, like that, because it is insoluble in alcohol. That is very, very important, my dear students. Okay. So go to the next one is then after the isolated gene transfer from the one generation to one gene to another gene or one organism to another organism. How it will be transferred? There are so many methods also. See that one? DNA is a hydrophobic molecule. Don't forget, my dear, hydrophobic. It is nothing but the water hatting. Okay, it will not mingle with the, it will not easily dissolve with the, okay, water, sorry, okay, water hatting. Okay, water hatting. Okay. Water hatting and cannot pass through the cell membrane. So this is very, very important. Gene transfer techniques are grouped into two main categories. So this is here, they may ask the question true or false. Direct question. DNA is a hydrophobic or hydrophilic. Hydrophobia, you know what about that? Rabies. When the rabies dog bite, what happened? The persons suffer. What happened now? By rabies, isn't it? As a result, what happened? They will not drink water. They fear about the which one, water molecules. That condition is called as hydrophobia. Likewise, the DNA is also what happened. It will not easily mingle with the water molecule. It is not easily dissolved. So hydrophobia molecules, water hatting molecule is there. So it has a two type of what is a technique, transfer technique, DNA, gene transfer, DNA technique. What is that? Vectorless or direct DNA transfer directly without any vector. We can easily introduce directly DNA into the host. Another one is called as vector mediated gene transfer. So the vector is also essential in between the gene transformations. This is also another category. First one, we are going to see that vectorless or direct DNA transfer. It has been further subdivided into two categories. One is called as physical gene transfer method and chemical gene transfer method. Okay, once again repeat for you. Gene transfer method is also, it has two types. Two types of main categories there. One is called as vectorless or direct DNA transfer. Another one is called as, what is that, vector mediated gene transfer. The vectorless or direct DNA transfer is also subdivided into two categories. One is called as physical and another one is called as chemical gene transformation. Okay, so we will see one by one in the vectorless or direct DNA transfer. So the physical method. What are the physical method? How it be transferred from the one organism to another organism? One of the first method is called as, what is that, electroporation. What is that meaning? So electroporation. Electro, so we have to use the electron. Poration, there is a small pore making process. Okay, just like what is that? So injection. Okay, see that electroporation is a process where electrically impulse of high field strength are used to a reversible permeabilize cell membrane to facilitate uptake of large molecule including DNA. So this is very, very important. How it by electroporation is also done, it's nothing but one of the process, right? Okay, in this field, what they are using, electric impulses are also they were used for the high field strength are used to reversible permeabilize, okay, cell membrane to facilitate uptake of large molecule including DNA. DNA molecule, if you want to upload the particular instruments, what you have to do, you are using electroporation technique with the electric impulses also. So as a result, what happens in electroporation, the external electric field is applied to the protoplast. This is very, very important because we are going to introduce the protoplast only, which changes the electric conductivity and the permeability of the cell membrane. Thus, the exogenous molecules found in the medium are taken up. Okay, exogenous means outside. What are the molecule present in it? It may be completely taken up. Only we need which one, protoplasm, not only the protoplasm. When you are using these type of things, inside there is a protoplasm is there, another cell membrane, another permeability, exogenous molecules also found it. Isn't it? All are comes out. See that one? Example, I will show you how it will be. Okay, just imagine. Suppose take an example. This is considered a cell, isn't it? Cell inside what happened, already in our little there is a nucleus is there. Okay. Inside other components also then present. Other components also. Other component, but what are the component? What are the component? Whatever it may be. Okay. So many components is there. Different colors also there. We need only this portion only. So we need this portion. How it will be isolated from, about one of the electroporation technique. So they were used a small syringe-like structure, structures like this, small syringe-like this structures. Okay, so here there is a small piston also is there. Okay, what happened now? It is used for the sucking process. Okay, sucking means it is nothing but the pulling process. So suppose when you are inserting the cell, so we are the piston is using. Okay, when we are using the piston, what happens? Inner, what are the molecule present in it? Inner, what are the molecule present in, including the high molecular weight DNA molecules also it insert inside. So all the molecules comes inside. After that, it may be used for the some other purpose. Okay, so this is very, very important. Okay, see that one. Okay, so which is taken up. Okay, so go to the next one is, see that one. This is considered Before pulses, during electric field, and after pulses. The cell membrane is this is the structure. Inside as well as outside is also there is a pressure is there, and plus charge and minus charge is also there. Introduce a gene or drugs, whatever it may be. Okay, we have to we are giving the electric field. Suppose when you are giving the electric field, what minus is there? So all the positive charge are again here. What happened now? Arrange in the manner. Okay, electric field. Induce a voltage across the cell membrane. Suppose it transfer from the minus to plus. So the minus or adhere on the surface of the outside the cell membrane. Inside, what are the positive charge molecule present? They are all present inside. After that, what happened now? After the pulse, cell heal with the gene drugs inside. So after the insertion, what happened now? This is the normal position. It comes normal position. Before pulse, during electric field, after the pulse. Okay, this is the electroporation technique. Okay. Next one is the particle bombardment. Microprojectile or biolistic method. This is very, very important, my dear student. Biolistic. What is the meaning of biolistic? Microprojectile and bombardment. Bombardment means what is meant? We are introducing the we are aim the target. Okay, with the help of the special equipment is called a gene gun. When you are using the gene gun, what happens? Automatically, it reaches the target exactly, isn't it? Likewise, the molecules also loaded into the gun, and also it exactly targetly we focused and also loaded. Okay, see that one. The technique of the particles bombardment, also known as biolistic, microprojectile, bombardment particle acceleration done by the GNA gun, otherwise called as gene gun. Okay, so the biolistic, microprojectile, and bombardment particles acceleration or gene gun, etc. is the most versatile and also effective way. It employs gunpowder driven device or helium driven particle of the gun. This is very, very important. Okay, so I will show you how the gene gun is also used, how they were used. Okay, the DNA bearing tungsten at gold particle. This is very, very important, my dear. This is also asked so many times. How many diameter? The gene gun is also made up of which metal? Like that, they were asked the question. This is also very, very important, my dear. Okay, the bearing tungsten and gold. Don't forget, this is very, very important, my dear. This is very, very important. Okay, so this is also very, very important. DNA coated onto the microscopic pellets is literally shot into the target cell. This is the aim. Okay, what we are going to do? Okay, we are focusing the target cells. Okay, so this is very, very important. The particle bombardment used to deliver drug fluorescent dyes, antigenic protein, etc. These are the important points. So once again, repeat for you. This is very, very important, my dear students. The particle bombardment, microprojectile, a biolistic method. The technique of the particle bombardment, also known as biolistic, microprojectile, bombardment, practical, particle accelerations, or gene gun, etc. The gene gun is the most versatile and the effective way. It employs gunpowder driven device or helium driven particle gun. Okay, this is also made up of, which one you know, the DNA bearing tungsten or gold particles 1 to 3 in diameter. Referred to as the microprojectile is accelerated into the living plant cell. Okay, DNA coated onto microscopic pellets. Pellets means, you know, it's loaded, so like as a pellet like structures. It may be loaded. So normally, you know, but gun is loaded with the what is that? Ammunitions, bullet. Here, pellet, difference. Literally shot into the target cells. After that, the particle bombardment used to deliver the drugs, fluorescent dyes, antigenic protein, etc. Okay, see that gene gun. So this is the structure of gene gun. Okay, see that plant cell. We are focusing. So here the loaded. Okay, here the loaded. What is a component of the DNA molecule of the sample 1? The plant is considered as a sample 2. Okay, so here the loaded one, it has a specific what is the tungsten and golden with a diameter 1 to 3 inch, isn't it? Diameter. So loader, and also it is also aimed to the target cell. So exactly, okay. So then only what happens. See that one. These are the other things also. That see that here the laboratory work they are doing. This is the structure of the gene gun. It is made up of inside there is a tungsten loaded is that okay. So and another one is also this one is also laboratory aspect they are doing in the lab. This is also plant. This is also another work. Okay, see that the bio-red is used to transfect cell in the culture and plant leaves. Plant leaves also directly injecting. Another one is what is that? The cell culture. These are the two parts there. One is animal part, and another one is a plant part. They were used for the cloning methodology by gene gun, particularly biolistic. That is very, very important. How it will be acted? See that this is the gene gun barrel. Okay, this is the barrel here. Plastic disc with the DNA coated gold particle. This is called as gold. But why we are introducing gold particle means we cannot able to visible which is the DNA molecules isolated from the specific interested gene. So we need some identification. This is the selected one from the superior quality like that. And also disc stabbed by the screen. So this is another part. DNA coated gold particle. It is also reached the target. Okay. So from this one loader, there is a disc stabbed by the screen, and also DNA coated gold plate particle is the it exactly reach the target. This is the target cells. Okay. Likewise, what happened? The gene gun is loaded from the particles and reach the target. Particle coated with the foreign gene release the foreign genes when enter into the target cell and integrated into the chromosomal DNA. This is very, very important. What happened inside, the integral part means what happened? The unition, recombination occurs within the molecules only. That is called as what is that, micro injection or biolistics or gene gun method. Here they were used so many times, which DNA coated. Okay, that is very, very important one. How it will be reaches, target cell. How it will be reached? Target cell and also integrated into the chromosomal DNA of the plant. Okay. So the next one is micro injection is also another method, very, very important one. It is also similar to that of the gene gun, but here there is a needle-like structure they were used. See that one? Micro injection, it is the injection of the DNA solution by micropipette into the cell. This is the exact meaning. Okay. The glass micro needle. So glass micro needle, it's made up of glass. The needle is also made up of very minute needle. Are also used for the transfer of the micromolecules into the cytoplasm or the nucleus of a recipient cell or protoplast, whatever it may be. Micromolecules means nothing but the DNA, RNA, or whatever, and which molecule we are going to introduce into the one plant to another plant or one animal to another animals. Micromolecules only. Okay, into the cytoplasm only. Okay, into the cytoplasm or the nucleus or the recipient cell of the protoplasm. Three parts they were selected. Nucleus maybe, sometimes it may be cytoplasm, sometimes it may be nucleus, sometimes it may be a protoplasm. These are the three part only we are using for the further process. So the next one, the recipient cell must be kept immobilized. Those who are receiving the foreign molecules, foreign DNA molecules, what happens in the cells? It will not move. Suppose target is moving means we cannot be able to aim the target, isn't it? That is very, very important. So the recipient cell must be kept immobilized in nature. Then only the micro injections micro injection process also successful. Otherwise, this micromolecules also it reaches the target cell. See that the cell is held against the solid support or the holding pipette, and micro needle containing the desired DNA is inserted into the cell. This is very, very important. Already told you. It's nothing but the loaded. Okay, already syringe. Okay, micro injection. we are uploading DNA molecule into the needle. Then we are selecting the target. The target cell is always what happening on immobilized nature. Then only it will be reaches exactly. Suppose if it is not moving, if it is moving means what happened now? It will not reach properly. The delivery of foreign DNA is done under the powerful microscope that is called as micromanipulator. We cannot able to see in naked eye. We are using the powerful microscope only. Generally, the molecules, whether it reached or not, we can easily identify it. See that one? So this is the holding pipette. Here the hold cell is there. Okay. This is cytoplasm. This is the center nucleus. This is called as micro injection loaded the foreign DNA molecules. Needle with the foreign DNA molecule. Exactly targeted. Here the nucleus only targeted, not cytoplasm. We are focused. So this is the holding pipette. The cell is immobilized. Then only that cell is captured. Okay, all holding with the help of the holding pipette. So it can be, this is called as coast cell. Okay, the nucleus is also visible. These are all examined under the microscope only. Which microscope? That microscope is called as what is that? Powerful microscope, micro manipulator. Don't forget, my dear. Okay, can visible in nature. That is very, very important. Powerful microscope. So after that, what happened? You know, it will be reaches like this. See that one? This is one of the original photograph taken by the lab, isn't it? See that this is the holder. Which one? Which holder is the immobilization holder? The male pronucleus. Here the nucleus is there. See that the visible nucleus is there. This is also nucleolus. Okay. So he also targeted. This is the needle. So this needle is also inserted exactly to the target cells. After that, what happened? You know, this is the pronucleus injection. So it is also inserted like this. Finally, it reaches the target cell. It incorporates with the coast DNA as well as a foreign DNA. So these are the process occurs within the short period. Okay, you have to do very carefully. Suppose sometimes what happens, you know, the cell membrane is also broken. So the inner side of the cell wall is also comes out. Inner side, what are the which one is the target cell? Target cell, cell bound or also comes out. So it is failure in nature. It will not successful. Okay. See here, they are given as a successful injection with the swollen pronucleus. That is very, very important. Swollen pronucleus. Okay, this is the nucleus, right? Okay, this is the needle, right? Okay. When you are inserting, okay, here the DNA molecules of foreign one is there. Here also the DNA molecule is there. This DNA molecule incorporated with the coast DNA molecule here in this part. See this part. So this is the successful one. Okay, after that we have to carefully remove the needle also. Okay, so that is very, very important. See the next one. So this is the exactly. Okay, so the holder. This is a cell. Inner side there is a nucleolus. This is the needle. So loaded molecules also transfer from the exactly to the target cell. So as a result, what happened? The foreign DNA molecules incorporate with the coast DNA molecule. It produces the new genomic structures. That is very, very important, my dear students. Okay. Okay. So another one is also called as liposome mediated transformation. Liposome mediated transformation. Otherwise called as lipofixins. We will finish it up to this level only today. We will see the remaining things on the next class, my dear. Okay. See that one. Liposomes, otherwise called as what is a lipid. Okay, just note it down. Liposome are artificial spherical lipid vesicles surrounded by a synthetic membrane. Spherical, artificial. Don't forget, artificial. Okay, so artificial. Of the phospholipid that imitate the structure of the biological membrane. So sometimes what happens? No, the cell wall is also surrounded by a synthetic membrane. It is made up of lipid molecule. That lipid is called as what is phospholipid, otherwise called as a combined protein, combined lipid molecule. Okay, compound lipid. We may call as which one is this one is called as a compound. Okay, compound lipids. Okay, sorry. Compound lipids that imitates the structure of the biological membrane. It is similar to that of the which one biological membrane structure, but it is made up of phospholipids. This can be induced to fuse with the protoplast using PEG. Don't forget, my dear, this is very, very important. Protoplasm fission. When you are going to study the protoplasm fission, the PEG is a very, very important. Otherwise, without PEG, what happens, there is no fusion takes place. The foreign DNA molecule as well as the host DNA, it will not united together. Without this chemicals, polyethylene glycol, simply called as PEG. Okay, the protoplasmic fusion, don't forget, in this part, we note it down. So this is also very, very important. Protoplasmic fusion. You have to isolate from the, which one, two different plant or animals, whatever it may be, plant or animals. Okay, plants or animals. Protoplasmic fusion. Two strand is also isolated from this technique we already know about. Cellulase or whatever the enzyme is were used, rupture the cell wall, isolate them. What is that? Protoplasm in different strands, and also you have to use the molecular scissor, cut which one is the desired one, and also united together same with the help of the PEG, polyethylene glycol. Here they will not use the ligase enzyme. They are use the polyethylene glycol as a what is a linking agent. Okay, so that is very, very important. Then DNA to be transferred into the packaged into the liposome in vitro and transferred to the target tissues. After that, the liposomes. After that, what happened to the liposomes? Introduced in vitro means lab. Okay, in vitro lab. Okay, and transferred to the target tissues. And the DNA entered to the protoplast due to the endocytosis of the liposomes. What is the meaning of this one? Endocytosis means it enter inside. Endocytosis. It also easily enter inside. Okay. So today we will finish it up to this level, my dear. So remaining things we will see on the next classes. I will summarize all those things simply. See that one. What are the things we will discuss? So DNA extraction. DNA extraction, you have to do the three things: is the rupture of the cell membrane and the separation of the nucleic acid and purification of the nucleic acid. Next, we were used some of those buffer containing detergent that is called sodium toadyl sulfate. Okay, so then only the molecule easily separated, and then not only that, and also protein molecule and also RNA molecule and also polysaccharide and also lipid also we can easily isolate it by this method. And the next one is what happened now after the breakdown. What happened now? Breaking of the DNA molecule with the help of some electric current is also used for this purpose. So then what happened now? That lysosome and also ethylene diamine tetraacetate is also used for this process for the breaking of cell wall. And then not only that, and it has a specific properties. For example, plant cell is also made up of cellulose, so that it can be easily broken by the cellulase enzyme. Okay, suppose if it is a fungus, means that it is also the fungus cell wall is made up of chitin, so the chitinase enzyme used for the breaking of cell wall. Another one is ethyl diamine tetraacetic acid is also one of the calcium chelating enzyme. It is also easily bind with the cell membrane. As a result, what happens? It maybe loses their ability, so it can be easily what happened. The DNA molecules also comes out. And another one is called as SDS, sodium toadyl sulfate. It is also used for the denaturation of the lipid molecules. And enzyme protease is also very, very important for the activity of the protein. Okay, for the inside of the cell wall. And then RNA molecules, about the RNA molecules means ribonuclease is used for the enzyme, used for the RNA activities. And not only that, after that, what happened? You have to use a chilled ethanol. So then only the DNA molecule undergo this precipitation process, precipitation. After the precipitation, what happened? You have to isolate because the DNA molecule is not easily dissolved with them or insoluble with the DNA in alcohol, ethanol. Okay. So the DNA transfer method already it is nothing but the hydrophobic molecules. Already water adding process is there. It has two types of transfer technique is there. One is called as vectorless method. Another one is called as vector mediated. Vectorless is called as a direct method. Vectorless is vector mediated means in between there is a vector is also there. The vectorless or direct DNA also further divided into two types. One is called as physical. Another one is called as chemicals. Physical method, there is a electroporation. And another one is called as what is that, the particle bombardment microprojectile or bialistic method. And third one is called as what is that, and the micro injection method. So these are the things. So today we saw it. And the last one is liposome mediated transformation, lipofiction. Okay, these are the things already. Now, in this class, we were discussed. The remaining things we will see in the next classes, my dear students. So thank you so much, my dear. Dhanalakshmi, are you
Thank you, sir.
right?
Yes,
Wrong?
sir.
Carry on, just to go through it. You know earlier about when you are going to attend the exam, May 7th, isn't
Yes, sir.
it?
May 7th.
Please
Yes.
follow it. Please download all the questions. So 4 to 5 years question, you have to download it, including Ajay also. You download it and practice it daily, that question also, okay?
Yes, sir.
Okay, because for example, 2015, sorry, 2017 or 20, whatever it may, which question you are taken, so you have to practice it more than twice or thrice. Once you attend it, okay, how many mistakes is there? Just rectify it. Again, you attend it. So likewise, you have to practice in 3 times. It will be better for your things. Okay, my dear.
Okay,
Okay,
sir.
bye. Take care. Bye. Take care. Bye. Good night. Ma'am, I'm in class. I'm in class.